Hi, everyone. Thank you very much for being here. This is basically just a lab meeting with friends. And so my name is Cyril Versoza. I am an incoming fifth-year PhD candidate from Suzanne Pfeiffer's lab in Arizona State University. And today I have the pleasure to be sharing with you a research topic that I've been looking into on the insights into the landscape of structural variation in aye-ayes, or Dalbentonia madagascariensis. So just to give us a quick overview, there was a gong. So compared to single nucleotide variants, structural variants are mutations that are affected by a minimum of at least 50 base pairs. And within these structural variants, they come in a bunch of different flavors. So we have duplications, deletions, insertions, translocations, and inversions. And within this group, there's a subset called copy number variants. And these are mostly comprised of deletions and duplications. And the reason for this is because your deletions and duplications affect the total number of nucleotides in a given genome once their event happens. And so these structural variants are very important because they shape genome evolution and function. They are involved in phenotypic adaptation, and they contribute to disease. And they arise through a combination of different processes, but the main two things are you have your recombination-based mechanisms and your replication-based mechanisms. And so given its importance, there has been a lot of research into figuring out how to detect these events. And for my talk, I will be focusing on the computational approaches that work with short-read sequencing data. And for these short-read sequencing data tools, they come in 4 different strategies, and we'll go over them one by one. The first one is read count. So essentially, you look at the number of reads or your read depth in a given region, and if there's a dip in your read, that could be a deletion. If there's a spike in your read, that could be a duplication. The next one is your read pair. So instead of looking at the number of reads, you look at the orientation and the span with respect to the expected insert size. And so this means if you have a larger insert size, there could be something in there that shouldn't be, or if you have a shorter insert size, there's something there that's just gone. Next, we have split reads. So instead of looking at the number of reads and their orientation, you look at the reads themselves and if there's a gap in between them. Because obviously we don't want any gaps there. You want an intact read. And lastly, we have the de novo assembly method, which, as the name implies, you just create your de novo genome and compare that to a reference genome. And so, due to anthropocentric reasons, most structural variant studies have been focused on great apes and rhesus macaques, which is arguably the most used non-human primate in biomedical research. However, I am here before you today to hopefully highlight that there is some benefit in incorporating data from an outgroup. What outgroup, you might say? The aye-aye. So you might be thinking, Sai, what's an aye-aye? So you might be in one of three groups. You absolutely know what I'm talking about. You have never heard of an aye-aye before, or you might have seen it, but you just didn't know it yet. And so to give us some more insight about our Guinness-colored creatures, here we have a few. So, if you have seen Madagascar 1, 2, 3, or Penguins of Madagascar and All Hail King Julian, you have seen an aye-aye. Because, King Julian's political advisor and right-hand man is apparently an aye-aye. But in a more serious note, the aye-aye is the world's largest nocturnal primate, and they're the only member of the extant Dalbentonidae primate family, and they exhibit a lot of unique evolutionary traits. And so as you can see from the live action picture over there, we have 3 things to note. We have the fluffy ears, we have a long skeletal finger, and we have, well, it's not seen there, but they have ever-growing incisors. And these 3 things work in unison for when they eat. So essentially what happens is that they look for trees and they knock on it, and if they think there's something hollow, they then use their teeth to gnaw, and then there's a hole, they grab the whatever grub they can find using their long skeletal fingers. And that's how they eat. And lastly, given thanks to deforestation happening in Madagascar, the aye-aye is one of the 25 world's most endangered primates, according to the IUCN. And so, given this Hollywood superstar, you would think we would have a high quality reference genome, since we want high quality reference genomes to detect structural variants. Unfortunately, we do have publicly available ones, however, they're pretty fragmented. So to help fill this gap, we made one. And so as you can see, I'd like to highlight that compared to the release in 2012 and 2023, we have chromosome level scaffolds that account for all of their autosomes and sex chromosome. We have an N50 of about 1,000-fold improvement when comparing the 2012 release to our release, and an 8-fold improvement comparing the last year's release to this release. And so N50 is essentially a summary statistic that encapsulates the completeness of your genome, and so the bigger, the better, and we got bigger. Next is, I'd also like to highlight that compared to the releases from 2012 and 2023, we were able to have a data set of annotated genes, which will be very important if you wanted to look at the genomic context of these structural variants. And another thing to highlight is the Busco scores of our genomes, and it's a pretty good sign that at the genome level we're at 99%, and uniquely we have a transcript level which is at 96%. So all of these point to it's pretty good. And also I'd like to highlight that the reason why this is called a hybrid genome assembly is because we use 3 sequencing technologies. We use short reads, long reads, and Hi-C. And so now that we have this nice genome assembly, we can then use this to discover and filter out structural variants. And so we have the following pipeline. So we first mapped reads using BWMEM, and we used a trio of structural variant callers, namely Deli, Manta, and Lumpy. And from there, we wanted to get a consensus call set, where using Survivor, where we only retained structural variants that were called by 2 out of the 3. And from there, we then used Duphold to do depth-based annotation and filtering using BCF tools. And as I mentioned before, there have been no releases with annotated genes, but fortunately we do. And so we were able to look at the genomic association on where these structural variants lie. And lastly, we were able to look, we wanted to look for Mendelian violations using BCF tools and then manual curation using Samplot. And so here is just a summary of the different structural variants we were able to see. So we have deletions, duplications, inversions, and in a single insertion. And we also wanted to highlight the putative ALU element. And the reason why there is no scaffold 9, we did not forget. So scaffold 9 is the sex chromosome, and so these are all autosomal chromosomes. And so in total, we were able to identify 1,133 structural variants, with a majority of them being deletions. And just to give you a quick breakdown, deletions, we have around 1,000, 81 duplications, 51 inversions, and a single insertion. And it's a pretty good sign that these findings are in agreement with what has been shown in rhesus and humans, where a lot of the structural variants that they have detected are mainly biased towards deletions. And so on the right, we see 2 proportion plots. So the first one is essentially the SV types, and the bottom one is the number of affected bases. And so you might be thinking inversions only account for 4.5%. However, they account for a larger number of affected bases. And the reason for that is because inversions are longer than duplications and deletions in IIs. And so here we just have the different histograms of the length versus the number of counts. And we can see that the deletions and duplications are skewed towards shorter ones, while your inversions are skewed toward longer ones. And this is, I'd also like to point out and highlight that this has been shown in other organisms where most deletions and duplications are skewed towards shorter ones because they're more tolerated by organisms, given that they have they're shorter and they have less of an effect. And if we wanted to look at the genomic context, around 95% of our structural variants are categorized as modifiers, and these include your intronic and intergenic regions, while the rest of the 5% are subdivided into high, moderate, and low impact categories. And as we can see, given that we have a bias towards deletions, deletions are comprise the majority of the different categories, apart from the moderate impact, which has a lot more inversions. So far, so good. Are we still following? Okay. And so, as I mentioned, we then looked at the different structural variants in our chromosome level scaffolds, and we see that they are distributed across all of them, and we wanted to look at the correlation between chromosome level scaffold size and the number of variants. And we can see that they are very strongly correlated with the deletions, duplications, and inversions. We didn't really put the insertion because there wasn't a single one of them, and it'll only be a dot. And so, unfortunately, from our data set of 114 candidate de novo structural variants, you weren't able to find de novo SVs. I really wanted to see one. I manually created every single one of them. We were hoping to find one, because if we were able to find one, we would be able to compare the reported de novo structural variants in both rhesus and humans. And I believe this is mostly because of sampling, given that in a previous study from rhesus, they had a total of around 4,000 structural variants, and from those 4,000, they only had 8 de novo SVs. And they used like a much larger pedigree. So they had 32 individuals, well, we only have 14. And so just to wrap things up, we were able to generate a fully annotated chromosome-level hybrid de novo genome assembly. And across 14 individuals, we were able to find 1,133 structural variants. And we see that there's a bias towards deletions, which is in agreement that has been shown in both rhesus and humans. Structural variants are distributed across all chromosome-level scaffolds. And finally, no genuine de novo mutations were identified among 10 II trios. And so this would not have been possible without the guidance of my supervisor, Dr. Suzanne Pfeiffer, the rest of the Pfeiffer lab, the Jensen lab, and my dissertation committee. And so if you like this talk, our lab is looking for postdocs. And so if you're feeling you wanted to dabble in population genomics of non-human primates, please reach out to me or go to that website. There's a tab there that says open positions. And with that, am I going on time? I think so. Oh, yes. So I'd be happy to take some questions. Thank you. Yeah. Thank you.
So do you try to compare gene copy numbers between other non-human primates in this particular?
Yes. So we were able to look at I didn't include that, but we have data on the gene copy, the copy number variants, comparing it to rhesus and IIs, and we've seen that there's a lot more II copy number variants are longer than rhesus. Yeah.
I know that mutation in Manta Valley and they are considered as both, there is only both copy of the mutation they are completely allelic.
Mm.
And I found it always really it's really a black box of what they are determined and stuff. And I would like to know your thought about it. Do you think that those duplications are meaningful? Or?
See, that's a great question, because we have not looked at it yet, but I feel like it's like at that point, it's more of how you try to approach the question, and so hopefully for the future directions, we will be looking at those black box duplications. Okay, I think there should have been a ding, but thank you.
So I'll get started. I'm Lachlan. I'm from the University of Queensland in Australia. I'm kind of getting towards the end of my PhD, so this is some of the work that I've been doing over the last couple of years. And so mutations are a source of new alleles and genetic variation, and these mutations can have effects on both phenotypes and fitness. And so it's important for us to understand the distribution of these effects and some of the factors that might influence that distribution for our understanding of evolution, both of traits and of fitness. To kind of illustrate why we might expect the distribution of mutational effects to change, I'm going to use a fitness landscape model. So a fitness landscape shows the joint distribution between the values of a trait on the x-axis and the corresponding fitness values on y. And the value of the value of the trait that has the highest fitness, which is indicated by the dashed line up there, is called the fitness optimum. If we consider a population that carries most individuals that are kind of in the vicinity of that fitness optimum, so they're all, so the population is well adapted, we expect that the vast majority of mutations to be deleterious. So where's my mouse? So those arrows on the figure kind of indicate the effects of mutations, and it doesn't matter whether they increase or decrease the trait, they're both going to be deleterious on fitness. If we now consider a situation where there's like an environmental change, or maybe the population colonizes a new environment such that the fitness optimum changes, there's going to be a change to the distribution of mutational effects. We see that this class of mutations here that increases the trait value can now have beneficial effects on fitness in contrast to what they had before. So what these arguments kind of outline is how the distribution of mutational effects for fitness can change depending on the context that the population is experiencing. But these have kind of relied on this assumption that the effects of mutations on the trait value itself were unbiased in the sense that it was increases and decreases to the trait are equally as frequent. But this is an assumption that has kind of received limited empirical testing, and so it's kind of a case of if mutations for fitness kind of depend on what the current fitness is, then is it also possible that mutations for other traits also depend on the value of what that current trait is? So it's important for us to consider this for kind of two reasons. The first is that for traits that do influence fitness, any biases on the mutational effects on that trait are also likely to have biases on fitness. And populations also carry a lot of phenotypic diversity, so if we see that mutations kind of differ among different individuals with different trait values, then that could shape how traits further evolve. So using this population of lizards here as a kind of toy example, we can see that they've got phenotypic diversity for body size within that population, and if you kind of treat each one of those lizards as a different genetic background, we might be interested in whether there's a difference, for example, between those genetic backgrounds that are really small and those that are really large, or maybe alluding to some of the results I'm going to show you later, we might want to know if there's a difference between those that are more average size versus those that are a little bit more extreme. So just before I move on, I'm going to drill into this body size point a little bit more deeply as well. So body size is a particularly useful trait for this because firstly, many populations carry like extensive diversity for body size. And body size is also a trait that may influence fitness, but we still have a relatively poor understanding of how body size evolves. Meta-analyses have demonstrated that body size seems to be frequently under positive directional selection, yet there's very rarely any kinds of phenotypic change, which is called like evolutionary stasis. And underpinning these observations is some gaps in our understanding of the genetic basis of body size, particularly surrounding the mutational effects that it experiences. So this kind of makes body size an ideal trait for this kind of study, where we can both consider the effects of genetic backgrounds, the effects of the mutational effects among different genetic backgrounds, which could also help us to build some of that understanding of the genetic basis of body size. So to do this, we investigate mutation for body size in 5 different genetic backgrounds of an Australian vinegar fly, Drosophila serrata. The backgrounds were selected from the Drosophila serrata genome reference panel of lines, which were developed by some of our colleagues at the University of Queensland. And these are a set of inbred lines that are sampled from a natural population, and we expect them to be reflective of that population variation. The 5 backgrounds were selected with reference to the range of that body size distribution. So we've got some that are really small and really large, so those kinds of extreme phenotypes, and a couple that are more average. And they'll be labeled A to E henceforth, so if I refer to those letters, that's what I'm talking about. From each of these ancestors, we expanded them out into a panel of 60 replicated lines and then maintained them with brother-sister mating for 30 generations to accumulate mutations in the standard mutation accumulation design. And mutations accumulate kind of indicated by those colored bars in the schematic up there. And the full sibling mating design allows these mutations to fix relatively unhindered by the effects of selection, unless they have very, very large effects. Then, during the course of this experiment, we were measuring body size every 2 generations in order to capture gradual changes, both in size and variance, as a consequence of mutation. And it's these kind of gradual changes that I'm going to now show you the results for. So first, we asked if mutations were increasing the genetic variation for size. An increase in variation would indicate that mutations had actually occurred and were actually accumulating. And so what I'm going to show you is called the mutational variance, which is a per generation measure of the mutational contribution to genetic variation. So first, I'm going to quickly address that B background. You'll notice that there are no error bars there. Although I said we had panels of 60 lines for each genotype, that B genotype experienced a large number of line extinctions, meaning it has a much smaller sample size than our other genotypes did. And hence, we weren't able to get very reliable estimates for anything, and so I won't be talking about that background any further, but we will proceed with the rest of them. So now we can kind of address the more major pattern, which is this kind of symmetric distribution kind of around the genotype that was closest to the mean originally. Quite interestingly, that C genotype that was closest to the mean size originally had a mutational variance that we couldn't distinguish from 0, which was a little bit surprising considering we expect that an experiment of this length should have yielded some evidence of mutation. In contrast, those 2 extreme genotypes had the largest magnitude of mutational variance and were different from that C middle genotype, with the D one being intermediate. And what these results kind of suggest to us are twofold. So firstly, that C genotype was either not experiencing any mutations at all, or was robust to mutations in that the effects were, there were mutations occurring, but their effects were suppressed. And also that those genotypes that are more extreme in a population are potentially contributing more genetic variation to that population, which may influence how that trait evolves further in a population. So we've got evidence that there were mutations, at least in some of those genetic backgrounds. That's great. So then what is the effect on size actually going to be? We've got an increase in genetic variation, but we want to know if body size is then evolving or being influenced by those mutations. So we followed up by investigating what I'll call the mutational bias, but is essentially, was there a directional effect of those mutations as they accumulated, which would then manifest as directional changes on size. And I'll just quickly point out that bias is on that scale of millimeters, but these wings are very, very small to kind of give you a sense of how small these changes actually are. And for these results, I'll just be presenting those 3 genotypes, A, D, and E, so those ones for which we actually had evidence that there were mutations accumulating. So first, what we found was that the average direction of mutations also depended on the genetic background. So with that A background, that one that was the smallest size initially, had a bias in mutations that indicates that mutations were making that flies in that genotype larger on average. In contrast, though, the 2 genotypes that were above average size did not have any evidence of mutational bias, although it's potentially very weakly negative, but yeah, that's still a little bit equivocal. And it may suggest that mutations are kind of biasing body size back towards the mean of that distribution, but we, like I said, those results are a little bit equivocal, and we'd like to kind of drill into this a little bit further. And I'll just touch on some previous work kind of in this sphere, because these results were a little bit surprising to what we expected. A lot of work that's been done in nematodes seems to suggest that mutations for body size are kind of overwhelmingly in the negative direction, which we kind of a priori took as evidence, or kind of a priori took as like a suggestion as to why body size doesn't tend to evolve. But what we found is kind of opposite of that, is that the only bias we could find is positive. And we're kind of speculating that that could be an effect of genetic background from those nematode studies as well, but we just kind of don't really have the information to really pull any of that apart. And that's kind of all from me, so I'll just quickly summarize everything I've just talked about. So we found that mutational variance increased in magnitude as the genotypes became more extreme in size relative to the population distribution. which suggests that those more extreme genotypes are contributing more genetic variation to the population, and that perhaps there might be some robustness in those ones that are more average. And there's a weak pattern that may suggest that mutations are changing size back in the direction of the mean on average, but which may then limit how much more extreme populations can become. And then that obviously has implications on how body size can evolve over longer timescales. And so that's all from me. Thank you for listening. I'll just quickly acknowledge our lab staff and research assistants and volunteers. That I breezed through it very quickly, but that experiment was massive and took ages to do, so they were a great help. And I'm also just going to shamelessly advertise that I'm nearly finished with my PhD and looking for a postdoc. So if anybody is interested in my skill set, come and have a chat to me, or have a chat to me about the other work that I've done as part of my PhD. Thanks for listening. Do we have any questions? Yes.
Hi, Lachlan. That was so important that the study was done. I'm so glad somebody has done it.
Yeah.
Really exciting. A very minor question, I guess, is for the mutational variances, as you're talking about size, you sort of alluded to this a little bit when you did that direction of mutational bias.
Mm-hmm.
Did you also just keep body size in millimeters, or did you log transform it since you were starting with backgrounds that diverge so strongly in size? And so you want to make sure that your VM is a proportional VM.
Yeah, no, we haven't log transformed the body size.
I would suggest doing that just because we have big sizes that, right, with mutation, if you want to make sure you're looking at proportional phenotypes.
Yeah, okay, that's a good idea. I'll have a chat about that, yeah. No other questions? Well, I'll be around for the rest of the conference. Oh, sorry, yes?
Did you look at directional effects of these mutations on wing shape by any chance?
We did, and that's like a longer discussion because that's one of the other chapters of my PhD. So yeah, if you want to have a chat to me about that. Basically, just briefly, what we did with that was we got estimates of a whole bunch of the wing shape traits, and then we've also brought in other data sets from our lab so we can kind of like try to get at some environmental effects as well by contrasting to different microenvironments as well. So yeah, it is something that we have looked at. Cool. Perfect, thanks.
My name is Kimberly Moser. I'm from the University of Oklahoma. I am Gavin Woodruff's lab manager, and I'm going to introduce you to the wonderful world of frankenworms. What I'm going to be talking about is actually a body size in mutation, how I have created worms. Okay, basically I'm going over body size, worm introduction, mutagenesis experiment, marvelous mutants. I picked one, then I sequenced it, then I also was looking for possible lesions. We'll do the near isogenic lines I created at the same time, as well as initial sequencing that I did on those, and what I'm going to be working on next. So body size. Body size is insane. There are 21 orders of magnitude of body size on our planet. They go from itty bitty 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 microbes all the way up to the blue whale. I'm not going to emphasize this too much because I'm sure as biologists we are aware. Caenorhabditis elegans, that is a model system in the nematode world, and you're going to get a brief introduction into worms if you're not worm people already. C. elegans is a model system. It's well known in nematode studies. Maybe you've heard of it a little bit in other biological studies. It is tiny. It's about 1 millimeter in size. It can be found worldwide, and it lives in detritus, rotten fruits, rotten flowers, and such like that. We work with Caenorhabditis inopinata. Caenorhabditis inopinata is the sister species to C. elegans, and most striking feature immediately, as you notice up here on the screen, is that it is almost twice the size of C. elegans, yet it is the closest relative of this species. It also develops much slower. A C. elegans worm will go from egg to reproductive adult in about 3 days. The Caenorhabditis inopinata, it takes about 4 days for it to go from a little bitty egg all the way up to reproductive adult. Now, they're also ecologically diverse. They are found in one area of the world at this time that we are aware of. They come from the Asiatic Isles like Taiwan, Japan. They also come from Indonesia. They also live in one particular fig. So they live in fresh figs, not rotting fruit. They are also dispersed using figs and their wasps, which is another amazing biological system that is fairly well known. Here we also have more body size. The vector size of the C. elegans worms are slugs, snails, centipedes, beetles. The C. inopenata is just a teeny tiny 1 millimeter size wasp. How do they get into the figs? Well, the wasps carry their dower form, which is a form that does not need to feed for up to 3 months. It injects them into the fig when it lays its eggs, and that's how they get into the figs. So we have now talked about C. inopenata briefly. It is living in fresh fruit, figs. It has wasps as their vector. Its sister species is C. elegans, which is a model system. And they are also gonochoristic, male-female, whereas C. elegans is hermaphroditic mostly. They also have an incredibly strange, repetitive background. C. elegans on the top, you notice on its chromosomes, a lot of the repetition is over on the arms, in the center not so much. On the bottom is C. inopenata. It's insane. It goes every which way at every which time. So I figured, okay, for my first project into the world of genetics is why not look for body size differences, because that was initially the first thing I noticed. So I did a forward mutagenesis screen. I read Sidney Brenner's paper on nematodes, and I did the EMS experiment, which is ethane methylsulfonate. And I figured, okay, why not? It's fun. So mutagenesis is one of the strongest and most powerful tools that we do have in biology. Many, many discoveries have been made using our forward mutagenesis experiments. This is where all the magic happens. These are NGM plates, nutrient growth media, which is what we actually grow our worms on. The spot in the middle is what they eat in the lab. We use Escherichia coli, OP50 strain. So this is where the magic happens. So I did the mutagenesis screen. I grew up 117 NGM plates. I rinsed them off. I put them in a tube. I used EMS. I shook them for a few hours, and then I plated 10 gravid females on each clean plate. Then I followed those for 3 days. I picked 493 worms. I put those gravid females on more plates, and because these are gonochoristic, we have to go out to the 3rd generation. So we allowed the F1 generation to lay their eggs, their babies to grow, mate, and then we screened the F3 population. After I screened the F3 population, I came up with 59 different weird-looking worms. I'm not a geneticist. I didn't even come from biology. To me, a worm is weird anyway. So what I found out is we had these little bitty baby cashew-looking worms. I mean, they're short and fat. We call those dumpies. And we had longer worms that looked like little threads that you could see with the eye. We also had some that had blisters on their outside body, blistered cuticles. We had a few that were rollers. These are really strange worms, and I wish I had time to show you a picture because it looks like they're rolling around in their own skin. Then we also had a few that had abnormal vulvas and abnormal tails. These were strange. I mean, 2 or 3 vulvas and tails with or without spikes. It's really, really cool, all the things that I found out. So what I did is I took the body size worms, which is what I'm more interested in, and after backbreeding and crossing and all that and all the death, I came up with 5 distinct lines. 2 were long lines, 3 were the short, dumpy, cashew-looking lines. So to show you what I mean by width and length being different, my little bitty worms were smaller than C. elegans. My longer worms were almost 3 times their size. The first 2 here you'll notice is the PD1074, which is C. elegans remanufactured line for wild type, and NKZ35, which is our Ishigaki wild type strain for C. inopenata. We have named our worms WOU, 1, 2, 3, etc., meaning worms of OU, which is the university I'm from. So we go WU 1, WU 2, WU 3, WU 11, WU 12, WU 13, and all that. That's how it goes. So we have the longer worms, which were like I said, almost 3 times as long. The shorter worms, which are smaller. Then we have the wide worms. I noticed immediately that all my mutants, no matter length or width, no matter what their length was, they were wider than even the wild types of their own species and the C. elegans. And I'll just show you some pictures. These are the wild type worms of C. elegans and C. inopenata. Then my frankenworms. These are my long ones, mutant number 4, mutant number 5, which is Wu12, Wu13. My dumpy mutants, which are the little cashew ones. There is a typo on the first one. Mutant 1 should be Wu1. Mutant 2, which is Wu2. Mutant 3, which is Wu3. We had to pick one because you don't want to work on all 5 at the same time. Whole genome sequencing can be expensive. So I picked one. We picked Wu11 because it was the most striking. We did whole genome sequencing to pinpoint the molecular lesion that may underlie the mutant phenotype. We compared their genes, critical for body size regulation in C. inopenata, with homologous genes in C. elegans, and I created near isogenic lines to help me pinpoint that lesion. So, the fraction of our reads aligning to the reference. Good, yes, that's a good start, yay. The number of sites with greater than 10x coverage when we compared them, well, we only got 10x coverage. We want more, so that's a future thing. We did ask for 30x coverage, and they owe us. So we will be getting more coverage. NKZ35, which is the wild type, we had 74,366,024 sites, or only 60.5% of the genome. And Wu11, which is our mutant, is 76,679,608 sites, which is 64% of their genome. C. inopenata assembly actually has 122,994,328 sites. Now, the number of non-reference homozygous sites in Wu11 and NKZ35. NKZ35, our wild type, has 1,109,891 sites. Wu11 is 958,666 sites. Then, see, of course, I already told you this, why I'm not going to repeat it. So we looked at the genes and compared them to the C. elegans homologs. The number of predicted high impact genotypes in our species, Wu11 had 1,604 sites. The wild type has 1,798 sites. Then the high impact genotype specific to Wu11 only, we had 248 sites in 233 protein coding areas. What kinds of genes were they? Well, we had 123 genes that had orthologs with C. elegans, either single copy or part of multi-member gene families. We had 85 genes that were C. inopenata specific genes with no homologs in either any Caenorhabditis species. We also had 39 genes that shared a single copy ortholog with C. elegans, and only one of these was a single copy ortholog that had a record of dumpy phenotype in C. elegans. And before you go, yeah, you found it. No, we didn't find it. It is one strong candidate. However, the F3 mapping and nil genotyping reveal that this particular lesion, this particular gene, is outside our mapping area. Unfortunately, this allele is not even in our mutant Wu11. So, sadly, that didn't work. Okay, so where are the lesions? Okay, this is actually mislabeled, my apologies. This is actually a bulk segregant analysis, not one-to-one analysis as it is labeled. My apologies for that. The y-axis actually measures the frequency of the Ishigaki, which is the NKZ35 strain, and our mapping strain, which is a Taiwan worm called PX723, which is the alternate allele. The blue is our wild type F3s, and the red is our mutant type F3s. Now, as the NKZ35 allele frequencies increases only on chromosome 4, we really assume that it is going to be on chromosome 4 on the left arm. Only in the mutants, this really suggests that the mutant allele is in this particular region. So, I'm sorry. This was the allele frequency. This is the allele frequency difference. So, we're pretty sure it's going to be there, we hope. And again, our F3 sequencing in log scale does show we actually have one little peak up there. We did try this gene. We did knock it out using CRISPR. Unfortunately, we did not get the phenotype. So it is not the high gene. Darn it. Okay, so then we created the near isogenic lines, trying to pinpoint even closer what we're looking for. Okay, so I made near isogenic lines. We also back crossed them like 20 times, and we had them sequenced. And this is what we came up with. We did each isogenic line twice, so we had 12 total sequencing events. And again, it's only on chromosome 4 where we see the difference. And it will take about 30 seconds for this next slide to load because there's a lot of dots. In the meantime, I guess I could do karaoke for you. Okay, here we go. As you can see, this is the only space on this particularly where it is all the way through every single one of our nils. We are assuming that our NOC3 or our Norman, Oklahoma allele for the body size change is going to be in this area. We are still looking at it. This we just received like a week before this conference, so this is brand new. We still haven't done all the data analysis of that. So what are we doing next? CRISPR, knock out more genes. Get more reads on the previous sample because they promised us 30. Complete analysis on our nil sequencing we need to go through. We're going to further back cross our mapping lines and our nils to further shrink that little area. We're going to categorize and investigate our remaining 4 mutants, and then I get to play with more, do more mutagenesis experiments because I'm a glutton for punishment. Okay, that is all you have from me. And I would like to thank everyone who is involved, Gavin Woodruff, who is my PI, the University of Oklahoma, NSF, and the Vice President for Research who funded all my wonderful Frankenstein experiments. And I will take any questions if you have any. Thank you. Yes?
So at the beginning of your sort of step-down analysis, you said you had about 1,400 predicted high-impact mutants, I guess?
Yes.
What's the approach to predicting whether one's going to be high-impact or not?
The bioinformatics was performed by Gavin Woodruff. I am still learning, so I do not have an answer for that question. But if you give me your email and stuff, I'm sure I can answer it at some point. Any other questions? All right, you're free, lunchtime. Thank you, everyone.